Prostate cancer is one of the most commonly diagnosed forms of cancer in Canadian men, with 1 in every 8 being diagnosed with it at some point in their life and 1 in every 27 dying from it. My research looks at the expression of different genes during the 4 developmental stages of prostate cancer. The first 2 stages involve the tumor being localized to the prostate, and this is known as a benign cancer. The last 2 stages involve the tumor spreading to nearby tissues, and this is known as malignant cancer. Many prostate cancer patients are at low risk of progression between benign and malignant stages, depending on the molecular taxonomy. When detected early enough, prostate cancer can be easily treated. However, the process is invasive, risky, and leaves the patient sterile. Since many men may never progress to late stage, it is often difficult to determine which treatment option would be best. My project has been done in collaboration with the computer science department, who have used computer algorithms on clinical databases. They have detected significantly altered expression levels of different gene isoforms at specific stages of prostate cancer. Gene isoforms can be alternatively spliced when certain sequences are inserted or deleted. This leads to a different protein, now known as a protein isoform. My research has focused on 2 alternatively spliced gene transcripts, WWP2 and STEAP2. The clinical databases suggest that these have significantly upregulated expression in prostate cancer patients in comparison to non-prostate cancer patients. I have already analyzed the expression of the identified gene transcripts in 2 late-stage prostate cancer cell lines, the LNCAP and DU145 lines. This was done by first extracting the RNA from the various cell lines and converting them to a DNA form. I then designed primers which bind to the specific DNA transcripts. This allows us to quantitatively detect the expression levels of the different gene transcripts during a polymerase chain reaction. My results from the experiments verified those of the clinical database, as both transcripts showed significant upregulation. To expand on these results, I am currently using a new series of cell lines developed by a research group at Michigan State University. The WPE1 series involves 4 lines which were artificially developed from a non-cancerous control line. These 4 lines each represent a different stage of prostate cancer. I am currently using the same techniques as before to test the expression of the different identified transcripts in these new cell lines. Gene isoform expression will vary between different stages and different aggression levels of cancer. As a result, I predict that the expression level of the different gene isoforms could be used as biomarkers to determine whether the tumor will progress or not and how quickly. This could be used to determine which treatment option would be best for a patient in order to minimize their risks and maximize their benefits. In addition, it would also hopefully allow us to develop more effective and targeted treatment options. Accurate verification of the clinical databases would also suggest that the computer algorithms could effectively be used to develop biomarkers for many different types of cancer.